Hi everybody, good evening and warm welcome to this lecture. In fact, I'm talking about the applications of biotechnology in medicines. So, biotechnology is very advanced nowadays, and we have met serious challenges successfully because of the advancement in biotechnology. If you take an example of diabetes and taking insulin at regular intervals to control diabetes. As you all know, the insulin, if it is not produced sufficiently in the body, then the glucose is not converted into glycogen and is not stored in the liver sufficiently. So, excess of sugar will be there or glucose will be there in the blood, which will cause the condition called diabetes mellitus and will impact or will have its own effects, damaging effects in all the body parts slowly. So that is why it is said that diabetes works like a slow poison, right? So control of diabetes is very essential, and one of the most sure method or way is having insulin. It is also the safest way, safest mode of treatment to control diabetes. But think of a situation where the human insulin is not available sufficiently. Then what will happen? It will be very difficult to control diabetes. You may think of a situation of taking insulin from the animal, from the animals, from the animal pancreas. But here also the animal extract insulin may not be as efficient as the human insulin. In addition to this antigen, which is the protein taken from animals, can produce side effects, reactions in the human body because of the antigen antibody reactions. Okay. Now, if you think about think of a situation where a bacteria can produce insulin, then this complex issue can be solved in a very simple way. You can grow these bacteria and can have insulin as much as you require. Fine. So earlier it was done. It was done by the animal extract, that is the extract of the pancreas, by slaughtering animals. They used to derive insulin, but this was not as efficacious as the human insulin, and also it produced some side reactions or side effects also. In fact, the insulin is having a structure of two polypeptide chains, linked by disulfide bonds. So this is the structure. Two polypeptide chains are there, A and B, right? And the maturation of insulin is very important because insulin is in the pro-hormone stage. Initially, the insulin is pro-hormone, that like proenzyme. So this pro-hormone stage is also having the third polypeptide chain, that is C peptide, which is free. But the challenge is in making a mature insulin which is not having C peptide. So the C peptide has to be removed. Now, how this, for the first time, how the normal human insulin was synthesized? Actually, in the year 1993, an American company called Eli Lilly, what it did actually, it made two DNA sequences corresponding to the two polypeptide chains of human insulin, A and B. and they introduced this, this DNA sequences in the plasmid of E. coli. In the plasmid means in the DNA of Escherichia coli, a bacteria. And what was done? What happened actually? In the body of E. coli, these two chains of human insulin were formed. Two chains. These polypeptide chains were formed. Now these polypeptide chains were separated, were extracted, okay, separately produced, and then they were joined by the disulfide bonds. Okay, students, so the human insulin was synthesized like this. The chains A and B were produced separately, extracted and combined, separated and extracted and combined by creating disulfide bonds to form human insulin. So you realize how biotechnology helped here to form human insulin. So I think I will restrict my talk here up to this only in this video. In my next videos, I will come up with your other parts of this chapter, that is gene therapy, etc. So I sincerely thank you for listening my videos, and I wish the very best for you all. Thank you so much. God bless you all.